thank my co-authors and collaborators on this work that I'm going to be talking about today. So Eric Buell, who's with Mount Hood Environmental, Michael Case, Emily Howe, Jamie Robertson, and Eileen Ettinger, who's here today, all of whom are with TNC, and Nick Van Buskirk, who's with WDFW. Can we advance? Great. Okay, so as you all well know, Pacific salmon are broadly in need of restoration action. This is due to a wide variety of factors, including habitat loss and degradation, fish passage barriers, fisheries practices, and increasingly also climate change. As you all also probably know, the salmon life cycle is really complex. So we know that what we do on land through our habitat restoration and land management can potentially benefit salmon by improving spawning habitat and juvenile survival. But we also know that climate change is going to put pressure on salmon really across the board at each stage of their life cycle. So it's difficult to evaluate how our land management practices might be affecting salmon without also taking climate change into account. Okay, I'm not sure. Maybe I'll do an unplug and a plug back in here. Apologies, folks online, just trying to, yeah. Okay. Desktop 2, share, and PowerPoint. All right, take 2. So yeah, and also there tends to be this perception, I think that, oh no, climate change, particularly climate-induced changes in ocean conditions, are such a huge driver that what we do on land can't really make much of an impact. So it's really important to try to understand the relative extent to which land-based actions can actually increase the resilience of salmon populations under climate change. So this is something TNC in Washington wanted to explore, not only generally, but also specifically out at their Ellsworth Creek Preserve. I don't think so, but it's possible it's...
Not working, Cole. See if you can get it to work with your machine, and then you can be the official pusher of buttons if it's Michelle.
Yeah. The cord is very heavy, and so it's putting it out.
Sorry, and that plug doesn't seem to work.
Maybe nothing. And yeah, we can try to
Take 3. So yes, this question of, what is the relative extent to which we can improve conditions for salmon in sort of the broader context of climate change. This is a question that TNC in Washington was interested not only generally, but also specifically out at their Ellsworth Creek Preserve, which is located near Willapa Bay in the southwest corner of the state here. So this region around Willapa Bay in coastal Washington has been managed pretty intensively for timber production over the last century or so. And so very few pockets of old growth forest remain in this region. This map here on the right, the little yellow patches indicate the few remaining patches of old growth forest in this area. Now, back in the year 2000, TNC in Washington had the opportunity to acquire the entirety of the Ellsworth Creek watershed, which is located down here at the south end of Willapa Bay. And by purchasing the entire watershed, they had the opportunity to preserve some of those remaining pockets of old growth forest. But they also acquired a watershed which had been heavily logged for decades outside of that one remaining patch. So a good portion of the watershed had been cut over a few times already, resulting in relatively young and dense and not particularly diverse or healthy forests. And this is what TNC in Washington would like to restore these forests to. So these are images from that remaining old growth pocket out at Ellsworth. You'll notice the really big old trees here in this image. There's also a diversity of tree sizes and species, canopy gaps, just generally a lot of structural heterogeneity, which creates good habitat sort of generally for biodiversity. And we also think healthy streams for salmon. So having access to an entire watershed offers TNC this unique opportunity to think big and try to answer some tough science and restoration questions. So for TNC, this is a living laboratory to test theories about restoration and management. Shortly after they acquired the watershed in 2000, TNC developed this experimental design with the goal of evaluating the effectiveness of forest restoration efforts out at Ellsworth. For the purposes of this project and this presentation, the details of this experimental design aren't particularly important because we don't have fine scale enough salmon data to actually tease out sort of sub-catchment responses to the different treatments. But just for context, there are these different designations across the watershed and different restoration treatments. So in the passive restoration subbasins, TNC is leaving the forest alone, letting it develop on its own, and not conducting any active forest restoration treatments, but they have fully decommissioned all of the roads in those subbasins. And then in the active restoration subbasins, the forests have been thinned. This improves growing conditions for remaining trees, provides opportunities for both tree and understory regeneration, and generally just diversifies the structure of the forest. So the question is, in the context of this type of watershed scale forest restoration, does this benefit spawning salmon? And if so, how? And again, what is the relative magnitude of this potential benefit within the broader context of ongoing climate change? So zooming out a bit from the Ellsworth Preserve, we decided to consider the broader Willapa Basin system in addressing this question. So WDFW monitors chum salmon populations in 3 index reaches within 3 watersheds across the basin. So first there's Ellsworth, where, as I just described, forest management over the last couple of decades has really focused on forest restoration for a variety of objectives. And then there are these other 2 watersheds, Lower Salmon Creek and Cannon River. These watersheds are owned primarily by industrial timber companies, and so understandably their management has been focused on timber production. So in our modeling, we're able to harness information and data from across these different contexts to try to understand how salmon might be responding to different types of forest management at this sort of watershed scale. So the specific research questions we wanted to address in this work were, first, how do land-based restoration strategies affect salmon populations within the context of climate change? And second, what is the potential for forest management specifically to increase the persistence of salmon populations under climate change? And we addressed these questions using an integrated population model for chum salmon in the Willapa Basin. So integrated population models, or IPMs, as you may very well know them, incorporate information from multiple data sources and importantly account for the uncertainty in those data sources. They are structured as hierarchical statistical models, similar to state space models, and they include nested models reflecting both process error and observation error. We fit our IPM to the 3 populations of Willapa chum using a newly developed R package called Salmon IPM. It was developed by Eric Buell and Mark Sherrill, and it fits IPMs to data from anadromous Pacific salmon populations using this hierarchical Bayesian framework implemented in STAN. And it was designed specifically to work with types of data that are commonly collected for Pacific salmon. Various model structures are available within the package, representing different life histories and data structures, as well as either independent sort of single population models or hierarchically pooled multi-population models. And users can specify stage-specific covariate effects using formula syntax within this package. So I put the QR code here to the GitHub. Check it out. It's a really awesome, powerful package and pretty easy to use. So just to walk you through the basic structure of the IPM that we used, I'm going to present just the single population version of this model, but just know that we used a slightly more complex multi-population version of this model to address our specific research questions. That being said, just to walk you through it, we assume that the unknown true number of recruits from brood year T are log normally distributed around a density-dependent spawner recruit function F. This can take the form of either a Beverton-Holtz or a Ricker model. We decided to use a Ricker model in our specific application because that seemed to fit our data a bit better. Within that function, there are parameters alpha representing the intrinsic productivity of the population or the relationship between spawners and recruits at low densities. And then Rmax represents either asymptotic or maximum recruitment, depending on which form of the spawner recruit function you're using. And then importantly, we have a temporally autocorrelated process error component. Next, we have cohort-specific age distributions. Here I'm showing ages 3, 4, and 5 because those are the ages at which chum tend to return to spawn. These are drawn from a multivariate logistic normal distribution, which also incorporates process error. Then finally, we have some sort of brood table accounting here. So the unknown true number of spawners in year T is the product of the total recruits from year T minus A multiplied by the proportion that return to spawn at age A. Summed over all of the age classes and then adjusted by the fishery mortality rate F. Environmental covariates can be incorporated into this model in a few different ways. Covariates that are expected to affect habitat quality are typically linked to alpha, or that intrinsic productivity parameter. Covariates expected to affect habitat quantity are typically linked to that Rmax, or maximum recruitment parameter. And then all other covariates are linked to annual recruit abundance, sort of post-density dependence outside of that spawner recruit function.
Yeah, so essentially equivalent to alpha.
Um,
Alpha is the density independent parameter. So is there really any difference between the first and third?
Yeah, so essentially, the covariate effects outside of density dependence would be like added to this mean here outside of the function.
Right, but alpha is a linear parameter, so isn't that equivalent? So, well, I've seen this come up before, and I'm always confused by why people distinguish between alpha and post-density dependence, because it's the same. Because once if you think of alpha, alpha is just a constant, right? So it's multiplying the density dependence function. Is there something that I'm missing? I think those two are indistinguishable.
Yeah, if it's an adult to adult model, then correct.
That's what I'm saying here, right?
Yeah, but so it's generally written because often then we're doing adult to small.
Right.
Density dependence is the first stage, and then yes.
Yeah. Okay. So there's for this model, those two things are essentially equivalent. Okay. Thanks.
Thank you. I learned something new.
Well, no. I would like to learn something new. See that economist.
And they.
Oh yeah, if density dependence is occurring, the multiple density, like Shiraz has got multiple density dependence functions, and then those two are totally different. But once if you only got adult to adult, it all falls in the they're indistinguishable.
Yeah.
Cool.
Yeah, and there are within the salmon IPM package and framework, there are spawner to spawner models and spawner to smolt to spawner versions of the model that you can implement as well. So yes, this is just a spawner to spawner model here. Okay, so yes, covariates can be incorporated in these different ways, and everything is indexed by brood year. So covariates get lagged by an appropriate lag time to reflect the sort of life stage at which we would expect they would be most impactful. And then finally, we have our observation model. So all of our observed salmon data are incorporated via this observation model. And without getting too deep into the details here, this includes likelihood components for both spawner abundance and age composition, sort of anchored on those unknown true states that we're estimating. And then just for simplicity, we assume that the mortality rate due to fisheries harvest is observed without error. Probably not very, but we don't really have a better way to approach it, unfortunately, with the data that we have.
It's for these systems is pretty solid. A very small fishery that has to be very well reported because most of these are ESAs.
Okay, so these are in river fisheries. You're not worried about ocean fisheries.
Yeah, I can't believe they're subjected to any.
Yeah. The only harvest that we accounted, there is commercial harvest for chum in the bay, and that's very well tracked. And so that's what we account for here. But yeah, kind of impossible to account for ocean harvest. Okay, so that's, I think, the end of the equations. Sorry, folks. Bringing it out to sort of a more conceptual overview of our modeling approach. So at the heart of our modeling effort here is this IPM for chum. The most critical inputs into this model are spawner abundances over time, age composition data for spawning adults, and then those fishery harvest rates. Then our other inputs, like I said, can include really any number of environmental covariates we want to consider. So in this project, we accounted for a number of different environmental covariates. We accounted for forest structure within those 3 index stream watersheds. We also accounted for estuary conditions within Willapa Bay, as well as ocean conditions off the Washington coast. And basin-wide hatchery releases. And I'll describe all of these inputs in a little bit more detail here in a moment. But after fitting the IPM to all of these different data sources, we're then able to identify whether and how the populations are responding to these different factors. And finally, we're able to use what the model learns about these relationships to project salmon population dynamics under different climate and forest management scenarios. So first, just taking a quick look at our salmon data, we're really fortunate that DFW has been monitoring the chum populations in Willapa Bay for many decades now. So this here is a time series of chum spawner abundance in those 3 index population watersheds since 1984. And this, again, is like the primary input into our IPM model. DFW also collects age composition data each spawning season, which is another really key input into this IPM. Then, as I mentioned, Willapa chum are harvested commercially in the bay, which DFW tracks annually as well. Those harvest rates are shown here, and these rates are derived from basin-wide harvest totals as well as basin-wide estimates of chum escapement. Next, taking a look at our covariates. First, to account for forest structure within these 3 watersheds over time, we use this really awesome data set produced by the Forest Service and Oregon State University. So they have developed what they call an old growth structural index, which is derived from both field and satellite data, and which reflects the abundance of large live trees, snags, and down wood, and the diversity of tree sizes. So these are all indicators of sort of old growthiness. And we also think these various factors are probably important for salmon habitat as well. This index is available wall to wall across our study area and on an annual basis. So it's a really powerful data set. Yeah.
Oh, is that the overlay of the?
Yes. Yeah. So our goal was to account for sort of watershed wide or watershed scale forest structure. So we summarize this index in a few different ways. Primarily, the primary way that we looked at this was by averaging it across the entirety of each of those 3 watersheds. But as a sort of sensitivity analysis to explore the effect of the spatial scale over which we are looking at this covariate, we also averaged it within stream buffers of varying widths, just as sort of a test of that. Getting into what some of these data look like. So each panel here represents a different spatial scale, moving from watershed scale on the far left to the sort of narrowest stream buffer on the far right. The 3 subbasins or index population watersheds are shown in the 3 colors here. So we have Ellsworth Creek, that's TNC's preserve, shown in the black. And you can see here in this graph that after the year 2000, which is when TNC acquired the watershed, forest structural complexity has been steadily increasing over time, which is really nice to see. Contrast that with the other 2 watersheds, where you can see there was a lot of clear-cut harvesting in the mid-2000s, which is showing up as these steep drops in forest structural complexity around that time and subsequent recovery. And then the trends are actually pretty similar, qualitatively similar across spatial scales. Although what's interesting to note is that forest structural complexity does tend to be higher on average as you sort of narrow in on the riparian zone. So next, we wanted to consider estuary conditions. And one really notable change that occurred in the estuarine habitat in Willapa Bay during our study period was the spread of Spartina, which is an invasive species of cordgrass and which expanded pretty rapidly throughout the estuary in the early part of our study period. Peaking around the year 2000. This map here on the left shows Willapa Bay, and all of the red areas are areas where Spartina had established by the year 2000. So it really was kind of everywhere across the estuary. Spartina, I should pop, there's the time series as well of Spartina acreage estimated throughout the estuary. So you can see this peak around that year. So Spartina, we think, could affect chum by competing with or altering native habitats, such as native eelgrass habitats, which juvenile chum do rely on for foraging and predator avoidance as they migrate out through the estuary during a really critical, period in their life cycle. Fortunately, some really concentrated and coordinated control efforts starting in the early 2000s pretty quickly got Spartina acreage under control in the estuary. So you can see that this sharp drop between about 2003 and 2008 And acreage is currently near zero and is expected to remain near zero, we think, with continued control efforts. This and onward is real data.
Yeah.
This is extrapolated backwards from sort of expansion rates that were estimated in this publication that I grabbed this figure from. So we don't have annual acreage rates prior to that year, but we have sort of the annual expansion factor.
Student supplies equally to each record.
It's just estuary wide. Yeah. Yes. Exactly. Yeah. We assume all three of those populations experience this sort of similarly, recognizing that they do pop out into the estuary in different locations. So I'm sure there are some differences there, but just for simplicity, we assumed one single time series covariate here for each of those populations. Next, there are 3 hatcheries in the Willapa Basin that release hatchery-reared juvenile chum. Hatchery chum have the potential to compete with wild populations for resources and therefore may negatively affect natural recruit abundance. On the other hand, hatchery releases also have the potential to inflate natural origin spawner counts if rates of straying are high. Unfortunately, in these systems, hatchery chum are not marked, so we actually don't have any way of separating the natural origin from the hatchery origin spawners in our modeling. So instead, to try to account for this potential effect of hatchery releases across the basin, we included this as another covariate in our model. Finally, getting to ocean conditions, sea surface temperature, as you can imagine, is expected to regulate salmon growth and survival during their marine life stages. Sea surface temperature off the Washington coast is what we're considering here, specifically in the springtime. Again, when chum are entering the ocean for the first time. It's been variable, as you can see, since the mid-80s, though, is projected to increase potentially quite a lot through the end of the century. Another important oceanographic factor that we wanted to consider was coastal upwelling, which is another strong driver of ecosystem productivity. And again, along the Washington coast in the springtime, a coastal upwelling has been variable since the mid-80s. It's also projected to increase through the end of the century, but really only slightly and not nearly to the extent to which temperature is expected to change. Yeah.
Sorry, equation question. When you're modeling these covariates, are you assuming that there's a hierarchical structure to the parameters? So in other words, although you've got a single index, the effect of upwelling on each population is different and coming from a joint distribution. Is that sort of how it's modeled? I'm just trying to think because when you assume the same parameter across space, that is a very strong assumption, whereas just a hierarchical distribution sort of makes more sense, but then you only got 3 populations, and so I'm trying to work out how you're doing this.
Yeah, so we assume the covariate effects are shared across populations. That is an assumption that we're making. So yeah, given that there are only 3 populations, that was the assumption that we made here.
That gets rid of the possibility that one population is actually affected more than.
Yes, exactly. Exactly. Yeah. Yeah.
Put it in our own little priors.
But she only got 3 populations.
Yeah.
Okay, so just to summarize all of the covariates that we considered here, again, we are accounting for forest structure within those 3 watersheds, which we're linking to alpha. Estuary conditions reflected by the acreage of Spartina across the estuary, which we're linking to our max. Hatchery releases and ocean conditions all linked to annual recruit abundance outside of that density dependent function. Okay, so now we fit the model. So just a quick look at some of our model fits for those three populations. Here we have the model estimated true number of spawners fit to the observed spawner abundance data. So our observed data are shown with the dots here. The solid line shows the posterior median estimate. And then the shaded intervals reflect those nested process error and observation error credible intervals. So this is just to show that the model does do a really nice job of capturing the trends that we're seeing in the observed data, which is great. And then interestingly, you probably picked up on this already, when we overlay these trends, you can see that there's pretty strong synchrony among the three populations. And in talking to people who know a lot more about chum than I do, the general consensus seems to be that this makes sense based on the life history of these species and the fact that they are likely responding to a lot of shared broad-scale drivers. Okay, so now getting into some of the population responses to those different habitat conditions or covariates in our model. So starting up at the top with sea surface temperature, we found a largely negative effect of sea surface temperature on recruit abundance within our three populations, which is what we expected to see. And a largely positive effect of coastal upwelling, again, on annual recruit abundance. So both of these findings align pretty well with what we expected going in. Next, we found a negative effect of Spartina acreage in Willapa Bay on RMAX for our three populations. The other way you can think of this, obviously, is we found a positive effect of the invasive species control efforts in the estuary that got this invasive cordgrass under control. Yes.
Quick question, effect size, really helpful. Did you Z-score? How did you, what does this apply to?
Yeah.
Yeah, because you've also got zeros for things like hatchery animals and stuff like that, which when I thought about Z-scoring that didn't sound like a good idea, so.
Oh, well, I did. Everything, all the covariates are centered and standardized, just
Okay.
mean standard deviation rescaled. Yeah. So the effect sizes are obviously comparable when the covariates are linked to the same part of the model, but like can't compare the effect size for RMAX here to these other two because they're interacting with different parts of the model.
Well, just knowing that one standard deviation is helpful.
Cool, yeah. And then the last thing on this slide here, so we found essentially no effect of hatchery releases on the natural population recruit abundance. This could be in part because hatchery releases in this basin are much lower than our estimates for natural production. So for that reason, they're not really all that likely to either compete with the natural populations or really substantially inflate our counts of those natural populations. Yeah.
Looked earlier that the hatchery releases and the Spartina were negatively correlated.
Mm-hmm.
And so how did you deal with that?
Yeah, they weren't, I mean, I did check for multicollinearity. They weren't substantially correlated to the point where it was likely to cause, issues with inference in this model. But yeah, that's a good observation. And then, yeah, the final covariate that we looked at here and sort of the motivation for a lot of this work is the effect of forest structural complexity. And so I'm going to show you the results for just two spatial scales. So we, fit the model separately to each of these to explore the sensitivity of our findings to these different spatial scales for this covariate. So starting with the watershed scale for a structural complexity, we found a positive effect of this covariate on the intrinsic productivity of our populations. And then moving to sort of the finest, most narrowly centered around riparian zone version of this covariate, we did find a slightly more pronounced effect of forest structural complexity when we sort of narrowed in on the riparian zone, but overall a consistent positive response across spatial scale.
How did you, what does 0.2 mean? Because you got 3 indices with different means. Did you Z-score within an index or across the whole thing?
Across the whole thing, yeah.
Okay, so just
Yeah.
higher a priori will give you a bigger effect. So if you got two flat trends, just because you got a higher mean is going to positively affect whatever that was that you put it on.
Okay.
Is that's?
I'd have to think about that. I'm sure you're
I
correct.
mean, these are small effects.
Yeah.
Deviation.
Yeah.
You talk about the ecology of this, like, I'm surprised that you did, I would think 50 would be the biggest bandwidth or buffer. Like, I thought that these effects would be just like direct shading on the river and like, you get 10 meters away and it's
Yeah.
impact. So what's happening 50 or 250 meters away from the river that affects 20?
Yeah, a lot of it has to do with effects on runoff and sedimentation as a result of different levels of forest cover and forest structure. And then importantly also, we have some clear-cut harvesting disturbance that is likely driving some of these trends too, again, through the same mechanism, right? So if you go in and you clear cut even in the upland, that's gonna have downstream effects in terms of runoff, stream flow, sediment inputs into those streams, even if it's not right up against the riparian zone.
So, I mean, does that suggest that you could try a different buffer that's like only upslope?
Yeah, I tried that. So, I mean, what you'll notice here, and what I ran into when trying to isolate just the effect of uplands, is that they're all highly correlated. So these trends in the uplands are highly correlated with the trends in the riparian zone. So it's really difficult to parse out how much of the response is coming from which part of the watershed because of that. Yeah. Great. Okay, so moving on to the final sort of stage of this modeling effort. What's great about this IPM framework is that fitting and projecting can all be done sort of simultaneously with this model. So in order to project future population dynamics, we simply augment our data set by including future projected values for all of our covariates. We then refit the model with the unknown future spawner and recruit abundances, essentially treated as missing data and estimated as additional parameters within the model. So to do this, we need to specify future values for all of our covariates. For future forest structure, we developed 2 future scenarios just for that Ellsworth Creek watershed. So now we're projecting for just one of those 3 populations. And that's because, quite simply, that's the watershed TNC has influence over, and so that's what we wanted to explore. So first we have an ecological forest management scenario in which we're just conservatively assuming that forest structural complexity stays at this maximum observed level. Obviously, we expect it will continue to increase just as forests develop and as we do continue restoration work in the watershed, but we don't really have a defensible way to anticipate what that trend might look like. And so we're keeping it simple and just keeping it as a flat line. Importantly, I think it's good to point out that this ecological forest management scenario could still include some amount of harvest or selective thinning of the forests for a variety of objectives. But the idea is that forest structural complexity across the watershed just generally stays pretty high. Compare that to our industrial harvest scenario, which assumes that starting in 2035, TNC decides to do a bunch of clear-cut harvesting in the preserve, causing forest structural complexity to drop sharply, and then subsequently recover following that same observed trajectory that we have here. And to be clear, TNC does not intend to do any clear-cut harvesting in the preserve, but we constructed these two scenarios really as simple bookends to try to quantify the extent to which choosing this more restoration-focused management strategy over something like industrial timber management might benefit salmon under climate change. Yeah.
Did like picking a start date of 2035 versus like another date have any influence?
Not a huge influence. Yeah, that's a good question. It was something we played around with a little bit. Yeah. For future climate, we considered 4 different climate scenarios represented here by these 4 different global climate model derived projections for sea surface temperature. These climate models differ in the extent to which sea surface temperature is projected to increase, as well as the sort of interannual variability of those projections. And then we're including just one future projection for coastal upwelling because we have a few different projections for upwelling, but they really don't differ much in terms of the extent to which upwelling is projected to change by the end of our projection period, which is 2080. Yeah.
Sorry, those are the actual projections or you're generating models?
Yep, these are actual projections from climate models. Yep.
Did you bias correct the?
Yes.
So those would be rescaled back to the mean, or because otherwise you got a huge jump in 2021.
I know, and I wish I had the full that jump sort of occurs outside of. It's weird that shows up. I wish I had the full trajectory there to show you, but it does look a little funny. It looks like it jumps up there, but that's because it does in the projections for whatever reason. Yeah, any other questions on the climate scenarios? Yeah. Oh, it's a business as usual. Yeah, these are all like RCP 8.5, is that right? It's the business as usual climate scenario. So yeah, these are our climate projections that we're using. And then I don't have figures for the other 2 covariates, but as far as Spartina acreage in the estuary, we're assuming that continues to stay near 0. And then we just assume hatchery releases stay near the sort of current basin-wide goal, which is 2.5 million released fry. Okay, so getting into our projections here for Ellsworth. So the 4 panels now represent those 4 different climate scenarios, and I'm going to be showing you 2 colors, which represent our 2 forest management scenarios here. So when I fill in this first panel, the first thing you'll obviously notice is that there are pretty wide uncertainty intervals here, which makes sense because of all the uncertainties in our data, as well as sort of compounding uncertainties in demographic processes, compounding over time. But what you'll also see is this difference that emerges between the ecological forest management scenario and the industrial harvest scenario, where the industrial harvest scenario sort of best estimate for our population projections does dip below that other scenario for some amount of time. And this pattern is sort of consistent across climate scenarios, though obviously shows up to varying degrees depending on the climate scenario we're considering. So this is useful to see visually in terms of what our population projections look like, but obviously difficult to interpret what these differences might mean in terms of actual population health. And so the final thing that we looked at was quasi-extinction probabilities for the population under these different scenarios. So here we're defining that as the probability that the running 4-year mean spawner abundance drops below 50 individuals at least once by the end of our projection period, which is 2080. And again, just to orient you, we have climate models along the x-axis and our 2 management scenarios represented by the 2 colors. So first, when I fill in the estimates from the industrial harvest scenario, you can see that there are pretty large differences that emerge across climate scenarios where quasi-extinction probabilities exceed 10% in the sort of most extreme warming scenario that we looked at. For reference, from a population viability standpoint, NOAA guidance suggests that quasi-extinction probabilities below 5% over long timeframes such as these are considered acceptable. And so we have 2 scenarios above that threshold and 2 below. Then when I fill in the estimates from the ecological forest management scenario, what you can see here is the buffering effect that this forest management strategy offers in terms of offsetting some of those negative effects of climate change. And importantly, in one of our climate scenarios, that buffering effect makes the difference between an acceptable and an unacceptable risk of population collapse. So wrapping it up with some takeaways, unless folks had questions on the final slide. Okay. Yeah, go for it.
Like the effect sizes were pretty small
Mhm.
and the differences in scenarios. I mean, it kind of seems to me like your covariates aren't waning a lot. So can you talk about how you like validated and like selected among different candidate models? And did you try one just without any of the covariates, and how did you do?
Yeah, we didn't do like a systematic model selection procedure because essentially we were interested in, we were making an assumption that these different factors that we're considering might potentially be important. And so we just went ahead and included those in the model, assuming that if they weren't important, that would show up in the effect sizes being close to 0. And then retaining them in the model, they would essentially have no effect on the projections because those effect sizes are.
I'm not sure about that, because the more covariates you have, the larger the probability you're in the tail. And you're caring about tail probabilities. So uncertainties can propagate particularly low, the ends of the distribution you care about a lot more than we would. So it's possible that a model without covariates would actually say things are actually quite good because you don't have the high variance basically. And this is propagating lots and lots of variance. So I'd pay some attention to model selection.
Yeah.
I think especially because like your climate variables, sea source temperature in particular, I think it was, the projection period is quite different than the period fit into, so you're extrapolating quite a bit with that one. I just think that, I would poke at it a little bit.
Yeah. I think, for us at the end of the day, our focus was less like getting these quasi-extinction probabilities correct and more looking at the relative differences among scenarios, which is why we chose to retain those covariates in the model that we were interested in. Yeah. Yes, Trevor.
I'm really intrigued to see how TNC looks at some views, these, because the previous slide about looked at that, I was like, well, it doesn't really make much difference to salmon, frankly. And this slide, I'm like, oh, okay.
Yeah.
Maybe.
Yeah, and I think, in the scheme of things, a quasi-extinction probability of 10% isn't all that high, but it could be meaningful for these populations in the long run. And chum are the most abundant species of salmon currently in Willapa Bay and are really sort of the cornerstone of the ecosystem there, as well as a lot of the commercial harvesting operations. So there is a lot of interest. and how our management might be affecting these species.
I think the other thing, sorry, speak for Tansfield. The other thing we think about is, that we know that chum are probably likely to be the least sensitive to some of these things of other salmon species, just because they spend little time in freshwater compared to the other species that are there. We use them in this analysis because those are the ones we have the best data for. Unfortunately, I don't have as good data on like Bovo or other salmon that might be more sensitive to some of these.
Yeah. Yeah.
You have to extrapolate that. Did you look at what would happen if you continue to improve the quality of the quarry?
I actually didn't, and I should. I'd be curious to explore some hypothetical scenarios where we do try to extrapolate that trend. I think I was hesitant to do that for a variety of factors, but I do think it's an important point in that because we're keeping that covariate flat. For this one scenario, we are likely underestimating some of the potential benefit of that management scenario. Yeah.
My thought was like similar thought process as you went through the slides as Trevor, but the slide where you showed like the scenarios, I noticed like the y-axis was pretty small, like it didn't go down to 0. So I wasn't sure like what, like I know the black line was the observed, but then the future lines were just kind of relative to that. But is kind of what Owen was saying is you probably need to poke it more to make sure the takeaway. I mean, because this slide, the takeaway is okay, they are doing like there is a difference between these scenarios, but another takeaway might be like, oh, if you harvest, not that big of a deal. But like, is there a world in which the recovery is slower because it's like happened before or something like that? I don't know that far as ecology, but like having the drop be lower or slower.
Yeah. I mean, another, this is a very sort of coarse scale way to go about probing this question of like, how does forest management affect salmon? Another avenue we thought about exploring, but couldn't quite get to make work, was using a more mechanistic approach to think about the forest structure effects on in-stream habitat via an eco-hydrologic model. With that, we could actually implement more sort of like nuanced forest management strategies, see how those translate into stream temperature, flow, sedimentation, things like that, input that as covariates into this model. Unfortunately, we had some troubles getting that eco-hydrologic model to work. And so we took a step back and said, okay, what if we just look at what forest structure's doing, recognizing that like there are all sorts of mechanisms there and probably lag times and things that we're not accounting for, but it's sort of our best shot at trying to capture some of these trends and relationships.
I do think the slide is super important, showing like there is a difference in scenarios, but maybe or thinking of ways to show that, like on this previous slide too, like what do the distinct lines look like not just the shading, but
the individual projection population. Sure. Yeah.
I mean, when I think about this, I go back to the data. And when I look at the data, I see for a forest, whatever that index was, quite a big difference in mean and trend between streams, right? But I also see a very highly correlated spawner index, right? So it's almost self-evident that you almost didn't need to do the model to know that the effect couldn't be large because you don't see a difference in the data, right? And most of the covariates were common across streams, which is why I asked my original question. So the only thing that you're seeing as distinguishing the streams is the coverage data, and you're not seeing much of a difference. So by definition, the coverage can't be making much of a difference because that's the one that's different. So I think it all hangs together, but in some sense, you have to ask the inverse question, which is, if there was an effect, what would you expect it to have seen in the data? And as far as I can see, I didn't see much of a difference. if you gave me those data, I couldn't tell you which is the stream that's benefit.
Sure. Yeah, I mean, I think that's part of the challenge again, because these species, as Eileen pointed out, are not spending a huge amount of time in freshwater. They're not as sensitive to freshwater conditions as other species might be. And they do tend to really respond to ocean conditions. So those like big broad scale drivers. But that was, exactly the challenge we were trying to poke at here is like, given that those broad scale drivers are really driving the ship here, are there still meaningful sort of like buffering effects? Can we turn the dial on that in slight but potentially like meaningful ways through our land management? And I do think these results indicate that is the case here. Yeah.
Sorry, I might have missed it, but what are the projections? What is the assumption on the fishing mortality?
On the fishing mortality? It was, oh yes, thank you. I had to dig deep in my brain for that. We made an assumption that fishing mortality rate would stay close to the sort of average that has been observed over the last 8 years or so in the basin. And that decision was made because of some policy changes that were implemented in 2015, after which we just assumed those policy changes would sort of stay in place into the future, and that harvest rates would sort of stay around the observed average over that time period moving forward. That's an assumption. We don't know what sort of policy changes might take place in the future. Yeah.
It's probably more realistic to assume that if the population was declining, you would have restrictions.
Right. Yeah. Mm-hmm.
There does seem to be some pretty big swings in catches
Yes.
when the population's also low.
Yeah. Yeah, and I think the policy, I'm probably going to get this wrong. I think the policy is like when they start to see declines, they limit catch to something like 10% of the total return. And I think the assumption we made moving forward was like 8%. So still below what that threshold would be if they were to implement restrictions in the future.
Again, this plot's not going to be affected by that because it's a common factor across all the population. So there's a lot of things that are going to scale out in a plot like this, but land use is the obvious one that won't, because it's a covariate that differs between the populations.
Takeaways. Okay. So this work, we believe, really does illustrate that land-based restoration strategies do have the potential to increase the probability of salmon population persistence under climate change. With a specific emphasis and focus in this study on forest management strategies that maintain and/or increase forest structural complexity. But also a continued estuarine restoration and spartina control in the bay. An important complement to all of this is in-stream habitat restoration. I didn't talk about this or focus on it in this research, but in more recent years, TNC has also been doing a lot of in-stream restoration out at Ellsworth. But we don't really have the data post in-stream restoration to try to account for that in our modeling. Another key takeaway here, forest structure can affect salmon at multiple spatial scales within watersheds. And this, again, is likely via effects on runoff, flow, sedimentation, shading, and woody inputs to streams over various timescales as forests develop. And I think this really highlights the importance of watershed scale forest management that does consider upland forests in addition to just the riparian zone. Finally, this research does add to the sort of growing body of evidence indicating that climate change has and will continue to put pressure on salmon populations. For that reason, it's important not only to address climate change, but also in the near term to pursue land-based restoration strategies that might offset some of those negative impacts. Before I wrap up, I wanted to mention some upcoming related work that we're getting underway. So myself and some of my co-authors on this are part of a working group funded by the Science for Nature and People Partnerships program. And our working group is focused on better understanding salmon responses to wildfire in the West and hopefully better integrating forest restoration efforts for fire resilience with salmon restoration efforts. So getting the forest people and the fish people to talk together a bit more. And as part of this working group, we're currently scoping out some research where we want to explore salmon responses to wildfire using a similar IPM and salmon IPM modeling framework. With that, thank you for your attention and your questions. I put a link here to the paper that we recently published on this work, which came out in Biological Conservation a couple months ago. So please do check it out. And yeah, I think we're at time, but I'm happy to stick around if folks want to chat.
Are there any super quick questions? I know folks have asked a lot before.
Yeah.
One quick comment, thank you. I wonder if, like, what do we know about where these fish go when they're out in the ocean? And because I kind of wonder if part of the issue of not picking up the facts is that, you know, it's not like, I don't think they sit on the shelf, right? And you had a slide with like a square, like the rectangle rather, like on the shelf from the Washington coast, like they, you know, take off and go into
Yeah.
Florida. I wonder if like maybe there's conditions that they do experience that your projections are not representative of.
Yeah, that's a really good point. I actually don't have a good sense of their movement or migration patterns once they're out in the ocean. We were really just targeting that like near shore ocean condition, assuming that when juveniles first get out into the ocean, that's going to be sort of the most sensitive, one of the more sensitive periods of their life cycle. And so that's the time period and sort of spatial scale we were considering in terms of ocean effects. But yeah, it's a good question. Beyond that, I'm not as sure.
Okay, we'll give another round of applause for Michelle. Thank you so much.